Well, thanks for inviting me. I heard Dan Rice's name mentioned. Is Dan here by any chance? Is he a member of this club? No? Okay. Oh, he's not here today. Okay. I can't see very well without my glasses, so everybody looks the same to me right now. I'm in a hurry to get here. I forgot my glasses too. But what I'll do is I'll give you a quick introduction to what GeneSeek is all about. It won't take more than about 30 minutes, the whole presentation. And then I'll open up for questions if you have some. And I think I have about 45 minutes, is that right? Okay. So just like Kim said, GeneSeek started here in Lincoln, Nebraska in 1998. And currently we are the market leader in genomic analysis for the ag and companion animal industry. And I'll sort of give you an idea of what genomics is and what we do with that. But in 1998 we started with one student intern in the technology part. This was started by myself and a fellow from the University of Nebraska, professor in animal science called Daniel Palm. So the two of us started that, and then today we are about 60 full-time employees. We are still in the technology park and hoping to move from there within the next year or so to our own building. Somewhere in town we're looking at a couple of facilities with about 8 PhDs and 2 DVMs. When we started, we were in a tiny little space in the tech park. We are about half of a building right now, about 15,000 square feet. And believe it or not, we process about 700,000 samples a year. You'll just, in the next few minutes, get an idea of what we do with those samples. And what we do with them primarily is what's known as high content SNP profiling. We also do marker assisted selection, identity management, and also a bit of clinical diagnostics on the same sample that people submit us. So the idea there is that it would be easy for somebody if they send a sample for DNA analysis to find out if that particular animal has a disease or anything like that. As you can see, everything is based on ag and companion animals. Now, it just gives you an idea at the bottom, all the pictures of all the type of animals we work with. We also work with a little bit with plants and trees. It's about 10% of our business, but just about the majority is animals, whether it's ag animal, production animal, or a companion animal. Okay, now what do these things mean? Just so that you know, genomics simply means the analysis of the entire genome or the entire DNA content of an individual, whether it's a human or an animal, makes no difference. If you look at single gene tests, that's exactly as it sounds. You're looking at a single gene. But the way we started the company was looking at single genes. Today, we do maybe 10% of what we do is looking at single genes. What we do primarily is looking at the entire genetic blueprint of an animal. Allows you to make breeding decisions. What is this animal worth? What can this animal produce down the road? Those kind of decisions. Should I keep them? Should I not keep them? For the traits that I'm interested in. Those kind of decisions is what will be used with this kind of information. Okay, in 2010 we were acquired by a publicly traded company called Neogen. They're based in Lansing, Michigan. So we are now part of Neogen. They're a fairly large global company, primarily focused on food security and animal health. So they do a large amount of testing for drug residues in milk, for example, and also everything from rat poison all the way to cleaners and disinfectants. You name it, there's a broad variety of products that Neogen sells. But they acquired us in 2010 with the idea that they wanted us to be part of their food security program. And I'll touch on that towards the end of this presentation, what I mean by food security. Okay, so what do we do? Everything we do is basically summarized in this one slide. From project management all the way to the different kind of tests we do are all shown here. We start from the very bottom, we talk to people who are interested in using our services, just figure out what they need to do, all the way from managing the samples, trying to get information out of those samples. And what happens is the kind of information we gather and the kind of platform we use is basically the three last columns. Everything from high density SNP profile. Well, just so that you know, what is a SNP? It's a single nucleotide polymorphism, or a change in a piece of DNA. Okay? It's very synonymous with mutation, but it's really not a mutation. We use platforms, which I'll show you in the next few slides. What does the Illumina machine look like? We also do our own panels. In the sense, what does that mean? Well, a company, for example, like PIC, how many of you are familiar with PIC? It's one of the largest pig breeding companies in the world. Now, PIC would use 300 or 400 different markers to select all the pigs that they would sell out to the market. So, for all the right traits that they're looking for, or somebody who wants to buy the trait is interested in a particular trait. But that's controlled by about 300 or 400 markers. We do all those work in label. And then we feed the information to them, and they know exactly what to do with that, and that's how they select that. Those are panels of markers. Panels could be everything from one marker all the way to 1 million markers. Okay? That's what those high density term means, up to 1 million. So that's how many we do. We also do full genome sequencing. We will do that very soon. We just got the machine in-house. That's what NGS is, next generation sequencing. So instead of looking at 1 million, you're looking at 3 billion bases, for example, in a cow. Okay? Same content as a human. And all the different diagnostic tests and the way we present them is also listed on the very edge of that slide. I know it's all a lot of technical information if you're not familiar with it, but I won't bore you with all the details, but basically boils down to the fact that information is provided to breeders and they can make breeding decisions. So what all sample shows up at the company? You name it, anything that you can think of that you can get a piece of DNA out gets mailed to us. Hair on cards, these two type of cards we call the middle, those are actually nasal swabs. Very affectionately referred in the lab as booger samples. And then those are blood cards. You can put semen on there, so we get semen also spot on those cards. We get ear tags, straws of semen, ear notches, blood, you name it. If we get ear tags, we have a machine that we just simply array those tags into, stick it into the machine, and it punches you those ear tags straight into a 96-well plate. Most of you may be familiar with the fact that there are ear tags that are out there that you can not only tag an animal, but you can get a sample as well. Okay, so that's all we need. We can get DNA out of it. We also sometimes get plant material punched out with that device that you can see somebody holding in their hand into a 96-well plate. I've already introduced the concept of a 96-well plate. Everything we do in the company revolves around a 96-well plate. In other words, we are not just simply dealing with 1 or 2 samples. Everything we do are in the thousands. Okay? So just to give you an idea, today we do about 15 to 20,000 samples of all types every week. That's how busy we are these days. So just a glimpse of the different labs. Sample starts over there in the DNA lab. It ends up either in the veterinary diagnostics group through some sort of a robotic system that isolates the DNA or the protein or RNA, doesn't matter what you're looking for. It eventually ends up in a 96-well plate. Okay? This is just to give you an idea of how we started. When we first started looking at single genes, we would take a picture of the DNA that shows you that over there, little bands on a gel, and somebody would sit down, look at the picture, and decide if there's a mutation there from the cutting pattern. Okay, that's how we started. And old technology. We also had some Lycor machines made right here in Lincoln, Nebraska by a company called Lycor. I used to work there. Those machines in the old days, those are about 10 years ago, pictures of all the machines clustered together in the company. What those data looks like, these are known as microsatellite markers. Most of you, if any of you have done parentage on cattle, these would be the markers that you would have used. Okay. So again, I hope I'm making some sense to you. Sometimes it may not, but that's what happens. This is how we started. The reason I'm showing you this information is that when we first started, if you were sending a sample in for your animal to be analyzed, it cost somewhere between $35 to maybe $100, $120. So the uptake was very limited. As Daniel Palm, my partner, used to say, it's the rich and curious farmers who are interested in sending samples. But we knew when we started the company that eventually the technology will be there that allows you to do a lot of information for about the same price. Okay? So as about 2004, things started changing, and the information content started going up. So for about the same price, now you could get hundreds of data points for a couple $100, or even sometimes $75 these days. You can get 1 million data points. So the information content, the amount of the content, and the value of the content changed. As the price went down, the information went up, but what you're getting has a lot more value than the money you just simply spent. And I'll show that in the next, in a few slides, what you do with EPD type of calculations. And some of you, I'm sure, are familiar with EPDs. Anyway, that's a nice shot of the DNA lab. That's the biggest group we have, about 20 people work in that group. All they do is isolate DNA in those 96-well plates. This is a shot of the, again, I like to show pictures because I can sit here and stand here and say all sorts of things about this is what we do, but it makes no sense until you see it. So that's what the veterinary lab looks like. The person that you're seeing closest to you is Suzanne Hinckley. She's a DVM. She runs that group. This is our mass spec lab. When I said SNP profiling, and you could do everything from 1 million to a single SNP. This machine, these machines that you're seeing there, we have about 5 mass specs in the lab. We can do somewhere about 1 to 400 data points on those machines. Okay, they're pretty expensive machines and they're very accurate, but the data sort of looks like little peaks there, but we can figure out at the end of a small molecule of DNA whether it's an A, T, G, or C, which is the 4 bases that make up DNA. And if those changes, then you know there's a disease or a problem with that animal. And that's basically what we use these systems for. So that's what that looks like. And those low density products, as we call, from 1 to about 400, we use those markers for parentage, breed composition analysis. For example, if you have a dog and you want to know, is my dog a certain breed or is it a mutt with many different breeds in it, we'll be able to tell you. Marker assisted selection, I'm sure some of you know that, or if you're looking for specific traits, if you have cattle and you're trying to find out if this animal is definitely a black animal or is it got a little bit of red in it, that'll be a trait. Or if you have pigs and you want to know if it's got the halothane mutation or the stress test, as you will refer to, we do that. That's part of the specific trait test that we do. Okay, so those are the low density profiles. This is the high density lab. Again, a picture of the Illumina lab, as we call it. Now it's completely in a different place. It's not an updated picture, but this gives you an idea. This is just a blown up picture of a glass slide that you would do if you are familiar with the high school biology class. You know what a glass slide looks like. Each of the strip is a band of small beads with DNA on it. So you can imagine how small that is. So this is, I think, 12 of them, 12 different animals being analyzed on a glass slide. So a blow-up version of that is shown there. Those little dots are all little beads with little bits of DNA on it. Each of those strips is querying 80,000 data points on an animal. Okay? Actually, in reality, it's in a redundancy of 20. Just to make sure you get the entire 80,000 data points back without any failure. So that gives you an idea. So that could be a million little dots that'll query then a million data points on an animal. Or it could be 7,000. So we do anywhere from 7,000 to a million. And that's, we call those, basically that's a chip. And we have our own chips, as well as we buy off the shelf from a company called Illumina. It just gives you an idea of the type of animals that we work with. Everything in black is off the shelf. Everything in red is what we have custom designed for ourselves. Just for your information, we sort of dominate this business in this world. When we first started, we didn't think we would ever get anywhere. But eventually, slowly, we started controlling this business. And this is how we did it, basically with these chips. So we get about 80% of the world's market. Everything comes through Lincoln, Nebraska. I always try to boast. Sorry, I have to do a little bit of boasting. But if there's an animal worth about $2,000 on this planet, there's a good chance, a 99% maybe chance that it's come through Lincoln, Nebraska and analyzed in our lab. Okay? Just from anywhere in the world. We get samples everywhere from South Africa to New Zealand, you name it, from Latin America a lot. And we do all sorts of animals. Now we have chips from chicken to fish. We just are ready to commercialize a Bos indicus chip for the Brazilian market. A Megamuga chip, as we call it, is for mice. And also a eucalyptus chip, which we are actually doing for the paper industry in Brazil. Okay, so here's a simple example of what you could do with this kind of information. Here's an example for a pipeline that we have developed within the company. A training population of animals, for example, here's an example of a Hereford herd. You take a sample, a hair sample, it comes to the lab, we get 80,000 data points out of it. We stick that into a prediction equation, a breed-specific prediction equation, and then it spits out an EPD, or expected progeny difference, or enhanced progeny difference, depends on who you're talking to. And some of you who have cattle know what I'm talking about. Gives you a little bit more detail here. Basically, EPDs are calculated by looking at progeny. It takes time. By the time you get 2 or 3 or 4 or 5 little progeny out of these animals, then you have a very good estimate of what that animal is worth. With genomics, instead of waiting 5 years, you can almost do it instantly. In fact, there are companies today, they take a little biopsy of the embryo, send it to us, and we figure out what the EPD is going to be of that animal before it's even born. Okay? So the EPD calculations with genomics, so that's basically genomic enhanced EPD. It's actually better than just pedigree-based EPDs, which is shown here in blue compared to genomic enhanced. So it gives you an idea of what. And then Dan is going to be in next week. It'll be very interesting because I've worked with Dan quite a bit. The dairy industry definitely changed because of this, the chip-based data. And I hope I'll also let Dan know to tell you a little bit about how we work together on a little test called A2, which is a beta casein mutation. And pretty much all his herd is now A2, okay? Because A2 is a mutation in beta casein, like I said, but supposedly that milk is better for you than with the small mutation in the beta-casein gene, which makes the gene called beta-casein A1. Most of the milk we get in this country, about half of, literally half of the milk would be A1. So you really would prefer to have A2 milk for you, because that's definitely the original version of the milk. So you can argue that's better. So Dan just decided he'll convert his herd into A2, and that's basically what he's done. And we helped him with that. Pretty much tested all his animals for it. So I'll let him tell you the story behind it instead of taking his thunder today. Okay, so that really enhances the value of the animal if you do genomic enhanced EPDs. Okay, I'll just simply just show you what we do, and then I'll just skip through that. I just want to get to the next slide. Just to give you an idea, we started here in Lincoln, Nebraska. Myself, like I told you, I was one of the founders of the company. One of the good things about being in Lincoln, and also one of the good things about being a crazy person like me, is that you see problems and you always want to come up with solutions. One of the problems we saw in the industry was that when people send us hair samples, they'll just put it in a little bag and just mail it to you. And when it's 10, 20 samples, it's not a big deal. You can take them and put them, get the hair follicles out to get DNA. But when thousands of them show up, it'll drive you crazy, because they fly all over the place. So we came up with this little device where you can stick the hair and seal it and send it to us. We can just simply punch through the device with the paper and the plastic, everything, and still get DNA out of it. Okay? So we came up with that. We got a couple patents on it. And I thought we'd sell about 50,000 of these over the next 5 years. We sold millions, literally millions of these cards. And we can't stop producing, shipping them all over the world. Little devices again. We make all sorts of things here. Lincoln has great machining companies. I don't know whether you are familiar with this, but Lincoln has great machine tool companies. For example, we have this machine we made here to punch ear tags, because there's a guy next door who could design it for us, and there are tons of companies around Lincoln who could make it for us. And we have a couple of these machines. We keep them going round the clock. Future, just to touch on it, I mentioned sequencing. This is what we are headed now to do. In the next few weeks, we'll be moving from our routine analysis of millions of genotypes to billions of genotypes. Basically genotyping by sequencing, or just completely sequencing an animal for the same price. Coming back to the price point again. The sequencing technology, I don't know whether you remember the Human Genome Project. It was a few $1 million to sequence one human being. Now we can do it for about 1,000. It's hopefully going to be $250 in the next 5 years. Tells you how much technology has changed. So, since you're the Ag Club, I thought I'll show the next 2 slides on a few points. So, as producers and well-wishers of the ag industry, I hope to leave you with something to think about. Food security, healthcare, water, renewable energy, and related, all of these are going to be significant problems, and also creates potential for things for us to do. Okay. As you know, world population is increasing, and demand for food is also increasing. Incredibly, the demand for protein is increasing. I showed you what all genomics can do to increase production, for example, in milk and protein. The big question is, can we sustain large animal production in the future for the protein demand of the world? Remember, as income increases, mostly in China and India or other countries around the world that are developing countries, the biggest purchase interest is for protein. So the demand for protein is going to go up. I'm a big fan of saying that sea farming of fish in big blocks in the sea is going to be one solution for it in the future. But I don't know about cattle being the main protein source in the future. I just wanted you to think about it. And food security becomes national security issues around the world. Okay? Just a quick Neogen slide, because Neogen is involved in all aspects of this. They are involved in food safety improvements. How do we reduce the impact on the environment? Also in agricultural output, both at the plant and the animal level. It's all part of food security. So I thought I'll leave you with that thought and then open up for questions.